Thank you, Martin. So I will tell you about a part of the BioPiglet project. That is a project founded by Innovation Fonden, where we want to use phages against enterotoxigenic E. coli to prevent, as a feed additive, to prevent post-weaning diarrhea. And I will skip part of the introduction thanks to Ans's presentation, so that's very convenient. But briefly, as you heard before, this is the main cause of post-weaning diarrhea in pigs, and it's a huge economic loss for the pig industry, and also for because it affects animal welfare. These E. coli are pathogenic E. coli, and we define them by the presence, by the exposure of two virulence factors that are the fimbria and the enterotoxins. And the fimbria are used by the bacteria to attach to the gut cells, and the enterotoxins are instead causing the release of electrolytes and water into the gut lumen, and this is causing the diarrhea in the pigs. And as you heard before, we are studying the diarrhea that is at the post-weaning age, so where the pigs are particularly sensitive because they also change the feed between the milk of the mother and the normal feed. These virulence factors are encoded by plasmids and prophages. So this is very interesting for us because the ETEC strains are actually very diverse in their genetic background. As I mentioned, the BioPiglet project aims to find a product that we can use in farms that are a very complex environment, and that we can use also for a long time. So we kind of feeling that we need to know a bit more about the phage-host interactions in these E. coli strains. From the literature, unfortunately, I could find only 27 phages that are infecting porcine enterotoxigenic E. coli, and we don't have really a full picture. So like from the first report, we have maybe more information about the ETEC, so about the bacteria. We have maybe information about the genomes or the fimbria and the toxins and some biological data, but we are missing the parts on the phages. And instead, the more recent reports are instead focusing a lot about the phages. So we have the full genomes of the phages, and we have the host range, but people don't report anymore which kind of fimbria and toxins the enterotoxigenic E. coli are encoding, and they don't sequence the strains. So it's a bit difficult to have a complete picture. For this reason, to have a well-characterized data set, we aim to first to assemble a well-described collection of ETEC strains, second to isolate and describe phages that are infecting ETEC, and then to identify host range determinants by combining the information about the ETEC strains and the phages. So first, the ETEC strains. So we have what is called the phage bio ETEC collection. And it's a collection of 84 ETEC strains from European pigs with post-weaning diarrhea that we collected between 2016 and 2021. And they are coming from Denmark, Belgium, Netherlands, Italy, and Greece. They are all sequenced, and they are all typed by PCR, and we also confirmed the typing of fimbria and toxins in silico. Most of them encode for F4 fimbria and F18 that are also very common, what we find in literature. And then from the enterotoxins point of view, also as reported in literature, we usually have more than one toxin. So we have usually a combination of different toxins, and there is a combination that is ST2LT that is extremely frequent. We also could type from the O type point of view these strains thanks to the in silico typing. And we found that we have 26 different O antigens. Most of them are O149. And this diversity is kind of expected because what I said before, like the fimbria and the toxins are shared on by plasmids and prophages, but the diversity of these genetic diversity of these strains is actually broad. The most common virotype, that is the combination of the Fimbria and enterotoxins, it's F4, at least in our collection, it's F4-SD2LT, and is often associated with the 0149 serotype. And this is important for later. So we were saying about the diversity of these strains, and I can say that to investigate the genomic background of this collection, we prepared this phylogenetic tree where we use not only our ETEC collection, but also the ECOR collection, that is a collection of E. coli strains that has been proposed in 1984 to represent the diversity of E. coli, and is still used as a reference. So you can see it represented here in this tree, and where you can see highlighted in a color, then it's an ECOR strain. So for example, in purple, in violet, you have like these ECOR strains falling into the phylogroup A. And for example, the one in the G are instead in pink. And you can see instead the ETEC strains that are falling into different phylogroups that are not highlighted. So, but we have ETEC strains falling into the A, B1, C, D, and D phylogroups. So very diverse, as we were expecting. And then I plotted in this phylogenetic tree also the virulence factors. So coming from, if we check the outer ring is the Fimbria, and then we have the toxins and the country. Yes, so as you can see, the Fimbria and the toxins don't cluster always with the phylogroups. And this is also expected because, as we were saying, they are encoded by plasmids, so they can be shared, and they move around among the strains. An observation that is very important for us is that all these virulence factors and most of the O antigens that we have in the ETEC collection is not represented in other E. coli collection, including the ECOR collection. So we are proposing the ETEC collection to be an extension of other E. coli collection. And we think that it would be very important both for groups of working with E. coli, but also with many groups, as we were seeing today, working with coliphages. So the second part was of course, the phage-related part. So we had to isolate some phages, and we did it using the same ETEC strains collection that I just presented. And we use different samples. We use gut and feces from piglets, from the same piglets that were affected by post-weaning diarrhea, but also wastewater, and also feces from farms with no report of post-weaning diarrhea. As media, we used a bit of everything, actually. We use LB, BHI, we tried without mucin, with mucin. And we use the samples both by direct spotting and with enrichment. And we found out that actually the enrichment were much better in general than the direct spotting. So we end up using the enrichment for the isolation of most of our stocks. So we reach more or less 180 stocks, but then when we started to sequence them and to test them on the strains, we realized that most of them were very similar. They seem very similar. So we actually end up working a lot on these three phages that are P21B, P22, and P102. And today I will talk more about one of them. But first, the host range of these three phages. Here you can see the same phylogenetic tree that I showed you before, with the ETEC strains and the ECOR strains. And you can see that I plotted the O antigen instead of the virulence factors this time. And then you can see stars where the three phages are infecting. So for example, P21B is infecting the strains where you can see a blue star. And P21B is our best phage, so it's infecting 1/3 of the collection. P22 is infecting a subset of these strains, and P102 is infecting only 12 strains, but interestingly is infecting some that P21B cannot infect, so they are kind of complementary. Another important observation that I think it's very clear here is that most of the infection is actually in this cluster of bacterial strains, of VTEC strains. I plotted the O type here because most of them are O149. So when I saw this, I thought, okay, then there is something related with the receptor. So P21B, as I said, is our best page. It has been isolated from a gut samples that was source of ETEC23, that is one of the ETEC strains, by enrichment of on another ETEC strain with LBM mucin. And it's an autography virida, but as T3 and T7. I'm saying that because T3 and T7 have been studied a lot, so they are very well annotated and very well studied. So it was very important for us to also help a bit the annotation of this page of P21B. So thanks to that, we can kind of recognize the genes probably involved in DNA metabolism and the structural genes and the packaging and the lysis genes. But they fall into different genera. So like P21B is structurally similar to T3 and T7, but is not similar enough from a nucleotide point of view to fall into the same genus. And indeed, P21B is a Berlin virus. And this is also important because we tested the T3 and T7 on the ETEC strains, and they cannot infect the ETEC strains. But this is kind of expected because we know from literature that T3 and T7 cannot infect when there is a O antigen. So, and our ETEC strains, as we were showing, most of them are O149, so they have an O antigen. So we investigated the receptor for P21B because obviously it was supposed to be different. So we have a collection of mutants in ET54. That is one of the strains that we are using also for the pig model. And they are mutants in known phage receptors. And thanks to that, as you can see here, P21B can infect most of the also the mutants, but it cannot infect anymore if we delete the WAAC, that is the transferase of the sugars in the inner and outer core. So of course, the O14, so P21B can infect despite the presence of an O antigen. And this is, of course, related probably to the tail fiber. So the O antigen contributes in some way to the phage host range, but is not probably the only determinants of that. So the problem of determining which genes are involved in the host range is much more complex than we were expecting or that we were hoping. So talking with our collaborators, Franklin and Pauline from the University of Southampton, they proposed to use, and they also run this functional enrichment analysis. And I will explain to you what you are seeing here. It's these are all the genes of our ETEC strain collection. And in black, you can see the ETEC genes that are conserved. And in gray, you can see the ETEC genes that are not conserved. And what they did is then to combine this information about the ETEC genes with the information about the host range. So in pink, you will see when the strains are resistant to the phages, and in green when the phage instead are sensitive. And by combining these two set of information, then we can find genes from the ETEC strains that may be important for the sensitivity to phages. And we have some preliminary results. We have some candidates, host range determinants that are statistically significant. We have 8 genes that are associated with infection and 20 genes that are associated with resistance, but we have to look more into each of them. So concluding, first, the phage bio ETAC collection, we think that this collection will help us to study not only the E. coli diversity, but also the coliphages diversity. So we think it's time that we also maybe share our collection to expand the diversity of the E-core collection. And then second, the O antigen and the receptor recognition, we know that they are important. We demonstrate that with the sequencing of our phages and also the test on the mutant collection, but they are not all the whole story. So by functional enrichment analysis, we identified other genes that are associated with phage resistance and infection, and we are going to validate some of them. I would like to thank all the people that have been working in this part of the project, and they are involved. So Lone, Seme, Martin, and Christopher from the phage bio group at the University of Copenhagen, and then Paulina and Franklin from the University of Southampton. But I would like also to thank all the people that have been working in the whole project, because the project is much bigger. So we have also a formulation part that is run by the Danish Technological Institute and the Easy Agricare, that is a company where they do feed additive. And then we have a piglet model for swine diarrhea, where we can actually control what is happening in each of the pigs. And we are going to test the phages hopefully soon this fall. And thank you for your attention.